In this video, we're going to continue with prokaryotic DNA replication, and we're going to start with the initiation of replication. So when the E. coli goes to replicate its genome, it will always start at the same place, and this is called the origin, or ORIC, and it's 245 base pairs, and it has a lot of, it has an AT-rich segment, which is roughly 45 base pairs. Now, this segment allows the DNA at ORIC, at the origin, to be separated, because the protein DNA A will come and bind at the origin at these AT-rich segments, and using ATP hydrolysis will separate your double-stranded DNA into single strands. And we need to use this ATP because we are breaking hydrogen bonds. We're breaking bonds, so we need to put energy in to break those bonds. When DNA A is at the ORIC, it will actually recruit two other complexes of DNA B. And DNA B is a hexamer. And if you imagine, if you imagine double-stranded DNA separating into two single strands, on one strand on each side, you're going to recruit this DNA B, because DNA B is a helicase, and a helicase will unwind this DNA. So you're going to continue to unzip your double-stranded DNA from both ends. So you're unzipping from both ends instead of one end, so it's going faster. And if we look at DNA B, that's it right here. The DNA here is in the middle. And we have 6 different colors, 1, 2, 3, 4, 5, 6. This is the hexameric complex, the 6 subunits of DNA B. And the way we're going to separate this DNA is through hydrolysis of any NTP. So DNA B can use ATP, CTP, GTP, and TTP to move this process forward. Now let's take a look at how DNA B actually unzips our genome. So here we have DNA B on our DNA, and the 6 different units are called A, B, C, D, E, and F. So E is kind of hidden behind here, but it's there. And the way that we're going to separate DNA is what's called a hand-over-hand method. So we have hydrolysis of an NTP. Here we're having it represented as ATP, but it can be any NTP. When we have hydrolysis at our topmost subunit, which is A here, A is going to disassociate from its neighbor. Here its neighbor is F. And when it does that, it's going to move 12 nucleotides further down the chain, and it's now going to be more closely associated with B. When A moves down the chain, the DNA unwinds by 2 nucleotides, and so the process is going to continue. F is going to hydrolyze an NTP. It's going to dissociate from E, move down to B with A, unzip 2 more nucleotides. E, same thing. Going to burn an NTP, move down DNA 12 nucleotides, attach to F, and unzip 2 more nucleotides. And it's going to do this over and over and over until the DNA is separated. Now, while you're doing this, the DNA is still a double helix further down, and so that helix is being twisted more and more and more. So you're building up this torsional stress, and if you didn't relieve the stress, the DNA could snap. You're forcing these bonds to be too tight. Luckily, there is a protein called Gyrase. So Gyrase, what it will do, it will go down to the DNA that's overwound, cut, unwind the double strand, and reseal. So it will just manually loosen the strain by unwinding the DNA and reseal it up. Now this DNA B, we said in the previous slide, it is recruited after DNA A, but it's loaded up by a protein called DNA C. Now when we're unzipping the genome, we don't want it to reform into double-stranded DNA. That would be a waste of our NTPs, our energy. And since, if we didn't do anything about it, since we have Watson-Crick bases, they would naturally want to reseal up. And so to prevent that, there's a protein called single-stranded binding protein, or SSB, that'll just cover all of our single-stranded DNA, actually physically preventing our DNA to reseal because it's being held by SSB. Now, SSB will hang on until a polymerase will come and kick it off. So it'll only be removed by a polymerase. You can see this is a U-shaped homotetramer, which means it has 4 subunits. All 4 subunits are the same. And the cleft, which binds our DNA, it's too narrow for double-stranded DNA. So only single-stranded DNA can fit, which is good because we don't want to accidentally seal double-stranded DNA closed. And the N-terminus, as you could imagine, has a lot of positively charged amino acids. Again, we're interacting with DNA, so this is kind of a trend. Anything that interacts with DNA, it's going to interact with the phosphate for the most part, and so you're going to have a lot of positively charged amino acids where you're interacting with that DNA. Now let's look at the actual replication mechanism for E. coli. Now we said this already, but we need a primer for any replication to start. We can't just start without anything there, so we need an RNA primer. And the primer is going to be added by a huge 600 kilodalton protein assembly called the primosome. And the primosome is shown in this yellow little bubble here. Now the primosome is made out of a lot of different proteins, so it's a complex. It's an idea. It's doing a job with a lot of different proteins. So you have your DNA B helicase that we talked about. Your primase is called DNA G, and we have 5 other subunits of this complex. And what happens is that this DNA B is using ATP to move, and this is driving the primosome on the lagging strand. So it's kind of pushing the primosome forward because remember, on the lagging strand, we need to keep putting new primers. We have to do this discontinuous replication, so we need a lot of primers. On the leading strand, we only need 1 primer and we're good, but on the lagging strand, we need to keep adding primers, so this DNA B is helping to move this primosome along so we can add new primers. So the main polymerase, and we mentioned this in the last video, is polymerase 3. So this will do the leading strand and most of the lagging strand. And if we look at the whole replisome, the whole protein complex that does replication for us, we see that we have 2 polymerase 3 enzymes, at least 2. Some organisms will have 3. And what's happening is that the leading strand will just be kind of straight, while the lagging strand is going to loop around because the polymerases, the poly3s, are all connected kind of together. They're connected through an intermediary, which means they can't move far apart. So to help with that restraint, the DNA itself is actually kind of bent over to look like a trombone. It's called the trombone model. So the poly3 will synthesize DNA on the leading strand. From our RNA primers, we're going to synthesize DNA, and that's where our Okazaki fragments is. But like we said in the previous video, our poly3 cannot remove the primers. It cannot do this Nick translation. So it will move all the way up to the Okazaki fragment. Then POL1 will come in and it will remove the RNA primers and fill that with DNA. And finally, ligase will seal up our last nick there. And now it is possible to remove these RNA primers through another protein, and RNase, or RNase H rather, shows up in like every organism, and it will remove RNA from any RNA DNA duplex. However, it cannot cut the last RNA molecule because this last RNA molecule has that phosphodiester bond to the DNA, so it can't remove that. So you still need PA1 to remove the very last RNA for you. And that's it for this video. I will see you in the next video.